Hi, I'm Valerie,
And I'm Leigha.
and we work in Dr. Alexa Veenema's Neurobiology Social Behavior Lab at Michigan State University. Today, we will be sharing a pilot study where we optimize new methods in preparation for performing a series of experiments to identify a neural pathway that regulates social play behavior in juvenile male and female rats.
Most juvenile mammalian species exhibit some form of social play behavior. This can be seen in human children, dogs, and laboratory rats. Previous research has shown that social play is a highly rewarding activity and that engaging in social play is important for developing social competence. Autism spectrum disorder, or ASD, is a neurodevelopmental disorder for which currently no treatments exist. Children with ASD exhibit reduced levels of social play behavior, and it has been suggested that this contributes to their lifelong impairments in social competence. Therefore, it is imperative that researchers investigate the neural networks that coordinate social play behavior to find possible treatments to restore social play in children with ASD.
To study the neural underpinnings of social play, we use an animal model. Specifically, we use juvenile rats. Previously, we found that neurons located in the lateral septum, or LS, that project to the ventral tegmental area, or VTA, are activated in response to the social play exposure in both male and female juvenile rats. Here, you can see a schematic of this neural pathway that we were trying to isolate and where the two brain regions in question are located in the brain. Our overall goal is to determine if the activation of the LS to VTA pathway is necessary for social play expression. We'll use designer receptors exclusively activated by designer drugs, also known as DREADS, to study this. DREADS is a relatively new technique in the field of neuroscience, which allows researchers to insert special excitatory or inhibitory receptors into the brain that can only be activated by administration of a special drug called CNO. In the series of experiments presented on this poster, we aim to pioneer the use of DREADS in juvenile rats as the first steps toward our ultimate research goal of determining the involvement of the LS to VTA pathway in the expression of social play. In order to express DREADS in the brain, the code for DREADS is incorporated in an AAV viral vector that also contains the code for a fluorescent protein. This viral vector infects neurons, and these neurons will then make DREADS as well as the fluorescent protein, which allows us to visualize the infected neurons. In order to inhibit the pathway from the LS to the VTA using DREADS, rats first received bilateral injections of the AAV, a control AAV, or one that codes for inhibitory DREADS, into the LS as shown in the images here. You can see in this image that the cell bodies of the infected neurons are green. Rats also received bilateral cannulation to the VTA, which allows us to inject the special drug CNO right into our desired brain region. The green fiber tracts in this image are axon terminals into the VTA, originating from the LS neurons that express the AAV. When we apply CNO, we are then inhibiting these specific LS axonal inputs to the VTA. In our pilot experiments, we examined factors that influence how well this AAV is expressed and how undergoing these surgery procedures affects baseline levels of social play behavior. The first factor we determined was which AAV serotype to use in our future studies. Different AAV serotypes may infect different types of neurons or have different infection efficacies. For our project, we compared two serotypes, AAV5 and AAV8, and we saw similar levels of expression in the LS for both.
In order to optimize the AAV infection time, we also had to decide what volume of the AAV we needed to inject into the LS. We tested 200 nanoliters and 400 nanoliters of the AAV as shown in these images here. The 200 nanoliters resulted in more precise infection of the LS, while 400 nanoliters expanded beyond the borders of the LS, so we will use 200 nanoliters going forward. The last factor we had to consider was the timeline of AAV infection. We tested AAV infection times of 9, 12, and 14 days, as seen in the images here. We saw infected neurons in the LS and infected fibers in the VTA at all 3 time points. Next, we needed to establish the ultimate surgery protocol. And you can see both options we tested on the diagram here. All experimental rats were weaned before arriving in our lab. Half of the rats received the LS and VTA surgeries at the same time on P23, and the other half of the rats received the LS surgery on P24 and the VTA surgery on P31. All experimental rats were exposed to social play tests with an age and sex-matched stimulus rat on P33 to P35. Rats receiving surgeries on 2 separate days had better post-operative outcomes than rats receiving both surgeries on one day. Additionally, the left part of this graph depicts that rats who received surgeries on 2 separate days, shown in black, had stable social play levels across days, while rats who received the surgeries on the same day, shown in gray, had unstable social play levels across the testing days. Therefore, we plan to use 2 surgeries on separate days approach moving forward. Lastly, we needed to confirm that CNO did not have any off-target effects on the behavior of juvenile rats. Rats infected with the control AAV received intra-VTA infusions of CNO or vehicle in a counterbalance over 20 minutes prior to social play testing on P34 and P35. Social play levels were similar under CNO and vehicle, which means that intra-CNO had no off-target effects on the expression of social play behavior, and therefore we can use CNO in our future experiments.
Going forward, by using the methods and parameters optimized in our pilot testing, we now plan to execute our experiments to determine whether activation of the LS to VTA pathway is necessary for social play. We predict that juvenile rats expressing DREADs that are inhibitory will significantly decrease their social play expression under CNO compared to their own behavior under vehicle, and compared to rats that receive the control AAV. This result would confirm our hypothesis that the activation of the LS-VTA pathway is in fact necessary for the expression of social play behavior.
Thank you so much for watching our presentation today. If you have any questions, please type them as a comment below this video and we will answer the questions in a timely manner.